Okay, so next speaker is climbing on the podium. Oops, I think I got the wrong picture for you.
No.
He's Cedric Notredame, so here's a real picture. And Cedric comes from Structural and Genetic Information Group in CNRS, Marseille, and is best known for serving excellent tea and coffee to the user community. He's also been indicted as a co-culprit in having written one of the two good books, Introduction in Bioinformatics. I'm sure he's going to ask me which one is the second one. Is that it? Well, I think it's Terry Sadwood book, Kevin. So basically, I put to you on GeoLink Heidelberg, the MBL, Lausanne ×2 and Marseille ×2, because you basically were in Lausanne twice during your career and you were in Marseille twice. And as Biolink, Des Higgins, of course, Jean-Michel Claverie and Philippe Bohar.
Well, thanks a lot for inviting me to this very exciting meeting and for being in so good company. I'm very honored. I also want to thank the organizer for giving me a chance to explore Brazil, and we found Varig every day in the hope that they will cancel our return flight. It's not funny for everybody. Yeah, anyway. So, as you can see here, the title I have chosen for this talk is Integrating Biological Information in Multiple Sequence Alignments. Now, this is very much about multiple sequence alignments and some of the stuff we have been doing with Des Higgins on T-Coffee and on the algorithm. And I'll be going around the details of the algorithm and all the latest applications. The word integrating is here as a token of a memory token about the word integrating that was everywhere in the proposal when I was a PhD student. There were very few grant proposals without the word integrating. Well, after all these years, now that I have a permanent position and that thanks to Des I have my PhD, I can admit that I do not really have a clue what the word integrating really means. Now, disintegrating I know, it's you take something big and you cut it in little pieces. But is integrating exactly the other way around? Is integrating exactly the contrary? I don't think so. Integrating carries the idea that biology is some sort of gigantic jigsaw where you only need a lot of patience to assemble all the bits and pieces. And in my opinion, more than patience, you'd need a pair of scissors to assemble such a jigsaw. This is why to the word integrating, I'd rather use the word confronting. In my opinion, a framework for analyzing biological data is a framework in which one can automatically and accurately confront bits and pieces of information, knowing that the bits and pieces of information generated by the biologist do not have to agree more than the biologists agree with each other. In my opinion, one of the best arena for organizing data fights, for making sure your data can fight with one another, is a multiple sequence alignment. Why is that? It's because in a multiple sequence alignment, you have sequences fighting with one another. You have all these sequences telling a different version of the same story. And the purpose of the alignment is to reconciliate all these lies and mistakes and omission in order to reconstruct something that looks like the original story. And I cannot think of a better data structure than a multiple sequence alignment to really have your data, your pieces of data fighting with one another. Now, many of you here are involved with support to users, and you all know that one of the problems in biology is that many people think the sequence alignment is solved. Now, if the sequence alignment problem was solved, this would mean that the ab initio folding of protein is solved as well, and that most important biological problems are solved. One of the problems we have with multiple alignments is that once you have generated your multiple alignment, people rarely use the multiple alignment or the alignment per se. They usually turn that alignment into a more sophisticated model. And they draw their biological conclusion on the most sophisticated model. Now, the problem is that even a mildly trained biologist will find it very easy to say, well, this portion of the alignment is weaker than that portion. But once the alignment has been turned into a flashy model, it becomes very difficult to convince the structuralists that they shouldn't use this model or handle it with care because it is not equally reliable. And that's one of the problems we have in sequence analysis. And that's why we must keep improving multiple sequence alignment and pairwise sequence alignment methods. Now, one of the reasons multiple sequence alignments have been putting bread on my table for quite a while now is because it's an extraordinarily complicated problem. It's a crossroad problem between biology, the actual definition of a good alignment. How can you mathematically define what is a good alignment, or whether this alignment is good and that alignment is not good? It's a problem that embeds most of the basics problem in biology. And the other aspect of the problem is computational. Assuming you know how to define your optimal alignment, and we usually define it through sequence similarity, trying to generate the alignment that maximizes similarity. But assuming you use this criteria or a more complex one, the question is to compute that alignment. And this happens to be what computer scientists call an NP-complete problem, a problem that cannot be solved through brute force computation, but that will need to be approached through the use of heuristics. Now, if you look at the literature, almost every optimization heuristics that exists has been thrown at the multiple sequence alignment problem. These days, most packages use the progressive alignment algorithm that Des was describing this morning. But I feel guilty about using sophisticated optimization techniques because my PhD was on genetic algorithms and multiple alignments. Now, here is one of the reasons why your progressive alignment strategy may fail. Let us consider the progressive algorithm like Des was describing it this morning, okay? Normally, if you do a progressive alignment, you will follow the order indicated by the tree, and you will start aligning A and B. And at that point, the program cluster, for instance, is smart and knows that it's a better idea to make a small mutation here and a terminal deletion rather than making a terminal insertion deletion, which is evolutionary costly. Now, the definition of a heuristic is that what looked good at the time appears to be a bad decision when you start adding extra sequences, sequence C and sequence D. And it appears that you cannot go back because you have already made that decision. And that's the real problem with a progressive algorithm. You may say, well, an iteration may solve it. In this case, yes. But if many more sequences are added, the mistakes will rapidly multiply so that another iteration will not save your multiple sequence alignment. Now, the way to address this problem is to try to ensure that these sequences, C and D, can start contributing some of their information to sequence A and B. And this is exactly the algorithm we developed for Tcopy. This is exactly what we try to do with Tcopy. How did we approach the problem? Let us simply consider all the pairwise alignments, A against B, A against C, A against D, and so on. If you consider all these alignments, how many alignments of A and B do you have here? Of course, you have the direct alignment, A against B, but you also have A against C plus C against B. This will be an indirect alignment of A and B through sequence C, and likewise through sequence D and all the other sequences. Now, it's very much like asking all the intermediate sequences what they think about the relationship between your sequences A and B. If you trust the alignment of A against C, and if you trust equally the alignment of C against B, this intermediate alignment will make sense. And if I can give you an image, in everyday life, we often use our own direct opinion, but we often also use indirect opinion. When you go to the doctor, that's exactly what you're doing. When you go to the garages, when you ask a plumber to fix your house, although this may not be the best image here. Now, if you accumulate all these triplets, all the intermediate sequences, you realize that you can now use this consistency in replacement of a standard substitution matrix. When aligning the two sequences in a standard substitution matrix, I will look up my PAM or my blossom, and I will therefore measure the cost for aligning this J with that J. In the case of the use of this triplet extension here, of what we call library extension in Tcopy, I will ask, how many times have I seen this residue with that residue in the direct or in the indirect alignments? Here, the answer will be 2. And that will be the cost associated with the alignment of my 2 residues. A summary of everything I have found in the library, in the extended library. One of the nice things with this library is that they totally separate the problem of actually computing the alignment and the problem of computing the pairwise alignments you put in the library. You can put in the library whatever alignments you're interested in. And in the original T-Coffee, we did use cluster W global alignments that were telling something about every residue in the data set. And we used L-aligned local alignments in order to generate the library. Now, how do you use this extended library? Well, you use it just like a regular substitution matrix. This is a progressive alignment strategy where you will start aligning the blue sequences, then the red sequences, then you will align these two profiles. In T-Coffee, rather than aligning the blue sequences with a blossom or a PAM matrix, we use the extended library to align the blue sequences. And the extended library, because it was extended using all the possible intermediate sequences, well, this library forces information of the red sequences into the alignment of the blue sequences. And we are therefore less likely to make a mistake when setting the gap that is here and when going further in the alignment. And this is why the information is more, there is a better use of the information in this consistency-based algorithm than there is in the regular progressive algorithm. Now, let me be very clear about consistency and where it comes from. We did not invent consistency. Goto started using it in the 80s. Martin Wingron also described it later. What we actually did was coupling the notion of consistency with the progressive algorithm. This was our contribution in T-Coffee. Now, the very nice thing about this consistency is that it really makes it possible to combine any kind of information you're interested in into your multiple sequence alignment, into a model that will be as consistent as possible with a multiple sequence alignment. And I will now show you two such applications, one in which we combine multiple sequence alignments into one single multiple alignment, and one in which we combine structural information with sequence information. The problem of combining many multiple alignments into one is something all of you involved in services have come across. Your users come to you and they say, what is the best method? And you can look at the current benchmark and say, well, last week math was the best method, so use math. But you know perfectly that this is not a satisfactory answer. Now, the satisfactory answer is that all the multiple sequence alignment package can sometimes be right and sometimes be wrong. So what you'd like to do in this situation is combine all your models into one, just like we do for structure prediction and gene prediction, using meta methods that are known to be more accurate. Now, how to proceed in the T-Coffee framework, that's fairly straightforward. Each individual alignment that we have here can very easily be turned into a library. And if you use 5 multiple sequence alignment packages, you will align your sequences using these 5 packages, turn each package into a library, combine the library, and compute the final alignment. If you consider all the existing multiple sequence alignment packages, it's quite complicated to define a satisfying protocol for combining all the methods and for deciding which method you will combine and which method you will not combine. A large part of this paper, of the M-Coffee paper that recently appeared in NAR, is all about choosing the right methods. Without going into very many detail, I'll just say that we ended up choosing one method per group, and we ended up combining this method. The tree that you see here is the tree we computed by comparing the alignment produced by each of these methods on a reference data set that was Armstrong. If you can compare the output of your method, you actually have a distance matrix. And if you have a distance matrix, you can turn that distance matrix into a tree. And that's what we did here. A striking property of this tree is that it very neatly separates all the algorithms according to the assembly algorithm. These are the progressive alignments. These are the one using die-align, a different assembly algorithm, and the one using POA, partial order alignment, a method using a different assembly algorithm. If you look into the progressive alignment category, you can see here all the consistency-based methods, except muscle, that shouldn't be here, but all the others are consistency-based methods. These are the fast Fourier transformé versions of math, and this is cluster W. So all the methods with a dot were the one we selected for M-Coffee 8, a method combining 8 multiple sequence alignment methods. And we have benchmarked mCoffee on the 3 most widely used reference data sets, that is Hamstrad, Prefab, and Bali-based, and came out with the conclusion that in most of these reference data sets, mCoffee was the method producing the alignments that are on average the most similar to the reference alignment. So on this reference data set on this benchmark, this will make mCoffee the most accurate method on average. Now, does this mean mCoffee always generates the best alignment? Of course not. And if you consider the choice of a multiple sequence alignment method as some sort of bet, like betting on a horse, well, if you simply ask the question, how many times on Hamstrad, for instance, how many times does mCoffee outperform all the other methods? We come with the figure 139, while in 65 cases, mCoffee 8 is the second best method or worse. If we average this on the entire data set, we can see here that, roughly speaking, mCoffee 8 will win on 1,000 data sets and will lose on 600 data sets. So this means you're not guaranteed to generate the best alignment, but you get good odds with you when you select mCoffee 8, which is exactly what we want to. Now, as you know, sequences are hard to align, especially when they are distantly related. And anytime you have structural information, I missed the slide. A nice side product of the mCoffee is that it delivers the alignment along with an estimation of the consistency of the final alignment with all the incoming alignment. So the regions that are in red here are in perfect agreement with all the individual alignments that you have combined together. And these are, as shown by Sonhammer in a recent paper in NAR, these regions are more likely to be correct than the regions that are not highly consistent across all the methods that are less likely to be correctly aligned. Now, as I was saying a minute ago, if you have structures, you want to use structures to help your sequence alignments. The reason is not, as you often read, that structures evolve slower than sequences. The reason is that the signal contained in the structures is more resilient than the signal contained in the sequences, and that some sequences can have evolved for such a long time that the sequences are now beyond recognition, although the structures clearly have some similarity. This is why one must use structural information whenever that is possible. Here is a standard data set on which you want to do this. You have here a group of sequences that are easy to align, another group of sequences that are reasonably easy to align. And if you want to make a profile-profile alignment, using structural information will be the best recipe here, the best way to make sure you get your alignment correct. Now, tCoffee is ideal for that purpose because all you need to do at this point is to use a structure alignment method that will produce structure-based sequence alignment that you will simply put in your library. You can do this, for instance, by adding the structure-based alignment in the pairwise global library. And this is what we do in Expresso. So tCoffee is notoriously slow, and Expresso does not stand for a fast version of tCoffee. It stands for expression, exprima, all the information contained in the structure. What we do in Expresso is that the user provides source sequences, normal sequences, and Expresso will blast these sequences against PDB in order to try to find what we call a template, what is called a template, that is a sequence with a known structure that can be used instead of the source sequence. This template must be very similar to the source sequence, must have 80% identity with 80% coverage. This is being very stringent. Once you have found the template, all you need to do is to align your template using your favorite method. And our favorite structural alignment method is SAP. So we produce a sequence-based, a structure-based alignment of the sequence template. And we then realign the source sequences with a template sequence. This is easy because the source and the templates are so similar. And then you simply have to remove the template so that you are left with an alignment of the sources that is actually a structure-based alignment. And this goes into the library for helping the computation of your final alignment. Now, we benchmarked this a while ago in the paper 3D Coffee by providing each multiple sequence alignment, each reference alignment with two structures in Homstrad, and we found that there was an improvement of 10 points, which is significant. Of course, when you start doing this, benchmarking starts being a problem because you do not have, you're actually producing your own structure-based alignments, and you're comparing these structure-based alignments with reference structure-based alignment. What does it mean when they are different? Does it mean your structure-based alignments are better than the reference, or does it mean that your alignments are not so good? This is a genuine benchmarking problem, and this is an issue I will address in a few ways at a small talk I'll be giving at the ISMB about the IRMSD, a measure we have developed for evaluating structure-based sequence alignment. Which takes me into the last section of this talk about the improvement of the evaluation. After many years spent looking at multiple sequence alignment methods and reference data set, I have to say I am not so happy about the way alignments are being benchmarked. If you look at the figures, I haven't them here, but I can show them later. Most of the methods perform about equally well outside of the twilight zone. When your sequences have more than 20% identity, you get about the same figures in terms of accuracy. Within the twilight zone, deep in the twilight zone, below 20%, the figures differ quite a lot. But it's hard to tell whether the problem lies with the reference structural alignment or with the methods. And what is very clear, and that was shown by Manfred Zippel some time ago, if your sequences are distantly related, there may be more than one sequence alignment that reflects the relationship between your structures. And this is the problem. The second problem I have with reference alignments is that at the end of the day, multiple sequence alignments are in most cases intermediate models used to build more complex, more sophisticated models from which biological conclusions are drawn. These can be homology models, profiles, 3D structures modeling, or annotation. And I feel that if we want to go further, especially in the twilight zone, we must start benchmarking not on the intermediate model, not on the alignment, but on its application. Is this possible? As far as profiles are concerned, that's fairly straightforward. A lot of literature has been dedicated to the benchmarking of profiles, and this should not be a problem. As far as trees are concerned, this is more tricky. Comparing trees is easy. Establishing reference trees is a matter of heated debate in biology. And as you know, you simply need to follow the literature. Every 2 years or every year, there is a major groundbreaking paper in Nature about the tree of life with a slightly different topology. And it goes on and on and on. So biologists find it difficult to agree on reference trees. As far as structures are concerned, CASP has put in place all the tools that are needed to check the accuracy of turning an alignment into a 3D model, and we should use this kind of things. And as far as functions are concerned, there is no framework for evaluating functional transfer, functional annotation, but I believe Swiss-Prot is ideally equipped to do this kind of things, and some benchmarks should be built upon Swiss-Prot to evaluate the accuracy of existing multiple sequence alignment methods. This brings me to the conclusion. One of the key ideas I have developed here is the notion of template-based multiple sequence alignments. The action of replacing a sequence with its structure is a very generic concept. You do not have to be restricted to structural information. You can use your sequence to map any information you're interested in onto the sequence. As long as you have a pairwise method that allows you to align your sequence according to the information you're interested in, you can put all these things into the T-Coffee bag and expect that information to be taken into account. We could replace sequences by profiles. We could replace sequences by sequences functionally annotated. And as long as we have the adequate software for aligning them, this will be fine in the T-Coffee framework. And this is the way we will go now. The second point was the need for a new evaluation procedure, which I believe has to take into account the application. I am not personally convinced that the same alignments will lead to the best profile, the best trees, and the best structural modeling. And finally, I want to insist on the idea that, in my opinion, integrating data is, whenever integrating sequence data, one must make sure that the framework is adequate for allowing the bits of data you're integrating to fight with one another. And this is the whole idea behind consistency and the measure of consistency. Here is a list of all the collaborators involved in this. This involved my collaborators in Marseille at the CNRS in the lab of Claverie, with Vladimir Saudek from Sanofi Aventis giving us a big hand. Sanofi is actually funding many of these people. The Irish wing of Ticofi is in the lab of Des Higgins, with Ola O'Sullivan behind most of the stuff done in 3D Coffee, and Jan Wallace have been done most of the stuff on M Coffee, and especially the very nice method tree I have shown here. Jan is around here. I can see him here, there. A big thank to the people in Lausanne, to Victor Yongenil and Brutoni Fleur from the SIB Vitality framework, who have provided us access to the Vitality machine and have helped porting all these programs, making available to the community a full Ticofi server where all the programs I have mentioned here are available online. And finally, a big thanks to the EPFL, to the lab of Roger Hirsch, who are giving me a great hand at tidying up the code and making it more efficient. Thanks to their help, Ticofi is about 7 times faster these days than it was 1.5 years ago, so they have helped a lot. One last word about this. This is actually carved in wood, and this was made in Salvador, which is something you can get in about 1 hour or 2, and you just draw a logo, and somebody carves it, and it's in the hotel, and I'll bring it along if some of you want to see it. Thanks for your attention.
So I can see there is already a question for Cedric.
Okay. When you said that integrating biological information should help improve the alignment, do you think that if you can also consider that the amino acid sequence derive from a nucleic acid sequence, that the fact that you consider the nucleic acid sequence alignment could also improve the amino acid sequence alignment? For instance, when you tried to differentiate in your example with Garfield, the last fat cat and fast, I mean the F and the L, there could be different reason for which the L was replaced by the F. If you can go down to the amino acid nucleic acid sequence, then maybe we can find what really happens.
That's a very good point. Now, okay, Garfield is a strange example, so I wouldn't fight on it. But in general, using DNA information rarely helps aligning proteins, because usually when proteins are so distantly related that they are hard to align, the nucleic sequences are even more distantly related. So by using the codons, you wouldn't get much help. This being said, your DNA sequences do not only contain codon information, they can only contain various features that are sometimes more evolutionarily conserved than the protein sequences are conserved themselves, and this can sometimes help anchoring the alignment. So using genomic information may be one kind of structures one will want to force into the alignment, just like we are forcing structural information into the alignment. Yes, I'd agree with you.
So are there any more questions? Okay, so the next, we'll start giving Cedric a